Hello, so today I'm going to be going over this paper titled Stress-Induced Low-Complexity RNA Activates Physiological Amyloidogenesis. So to begin with, I just want to really briefly introduce liquid-liquid phase separation. This just refers to the demixing of certain biological components and molecules within a cell from the cytoplasm. And so what happens when they demix is um, they start to create these kind of condensates. Sometimes they'll be called foci, granules, aggregates, all kinds of things. But what happens is they actually exhibit different material properties than when they don't undergo this transition. And for that reason, we kind of can consider them to be phase separated. Um, and people also sometimes refer to these condensates as being membraneless organelles. And that's because, within an organelle, what you're basically doing is creating a little compartment for where certain reactions are maximized and other reactions are excluded or limited. And that's basically what happens within these condensates, except they occur without a membrane. But rather, there's something innately within these molecules which is allowing them to exhibit this phase separated state. And we refer to this ability as multivalency. And so, what this means is under certain environmental stimuli and certain conditions, especially under stress, like heat changes or pH changes or nutrient deprivation, these molecules are really dynamic and they can change the way they function and their conformations and things like that. And that can help contribute to them being in this phase separated state. And oftentimes, like a good analogy for this phase separated state is like in a lava lamp. So you might consider the kind of clear fluid as being like the cytoplasm and these condensates as being the really opaque blobs, because the blobs are essentially within the fluid, but they've kind of separated and condensed together to seem a little bit separate from the fluid, but still within it. And yeah, um, and so there are a lot of things that we do know of and have already known of that we now consider phase separated condensates, and that includes the nucleolus, P granules, and protein aggregates. So just want to say that this is not, like we're discovering new things, but rather we're just looking at them in a different kind of approach and perspective and kind of uh, getting some advice from physicists and material scientists and things like that. And if you're kind of struggling to kind of figure out the kind of whole membraneless organelle thing, I like to kind of think of it as like a force field almost, because these molecules can innately kind of bring up these condensate barriers whenever they want to, but they especially occur when there is a certain stressor on the cell, and that's kind of like when supernatural characters kind of start to put up their force fields when they're against an opponent and feel in danger. So use that analogy if you like. And so also I just wanted to introduce some terms which will be used in the paper. So the first one is amyloid bodies. These are insoluble fibrous structure consisting of these aggregations of proteins that are arranged in beta sheets, and they're really hard and solid in a way. Um, there's kind of it's just like the definition explains. And then there's also low complexity RNA. So this is RNA transcripts which are transcribed from intergenic regions on ribosomal DNA, and they're mostly filled with repeats, and they don't really encode ribosomes or any other proteins or anything like that. And for that reason, a lot of times previously they were considered junk DNA, but in this paper, they're kind of hinting that, actually this isn't junk, but it may be relevant in this function of developing these amyloid bodies. And so that will begin with the first figure. So what these researchers notice is within these ribosomal intergenic regions, there are spaces which are actually well conserved in between humans and mice. And so as a geneticist, when you see a conservation of something in the genome, you're probably like, oh, there's positive selection because this is somehow, needs to be within the genome, and it's being conserved. And so there has to be some kind of function in it. And so what they find is this transcript from this low complexity region is really only expressed when the cell undergoes a certain stressor. In particular, they simulate a heat shock. Um, and when that happens, you get a lot of these transcripts around this 16 kilobase and 22 kilobase region where these RNAs are transcribed from. And then to the right here, you see that they do an RNA fish. So they're just adding a fluorescent probe to see where these RNA transcripts are. And what you find is when you do no treatment, so they don't apply any heat or anything, you don't see the fluorescence. So there's none of these RNA transcripts. But over here, you see that they have applied a heat shock and you see the fluorescence, which means these RNAs are being transcribed. And so that's already a really unique relationship. And then so what they also find is when you do the heat shock and you get the transcription of these low complexity RNAs, these foci start to develop within the nucleoli. So you see in this darker region, there are these lighter little circles, and these are these foci that begin to develop upon the transcription of these RNAs. And so what they begin to believe is there's probably some relationship happening that causes these foci to occur because of the RNA. And so to test that, what they do is siRNA. So they apply siRNA to these low complexity RNA, and what they find is these foci do not develop when you deplete this RNA. However, when you deplete a RNA which is high complexity, you still get these foci. So they're really kind of establishing that which is this SIP18 RNA. It's in the intergenic region, but it's actually high complexity. So you still get the foci. So they're really pinpointing that this relationship is really exclusive to these low complexity RNAs. And so what they believe is this low complexity region is interacting with protein to kind of have this interaction that recruits them and makes them condense up. And so in the next figure, that's what they find. So they find that there's this amyloid converting motif, which is really similar to this already known beta amyloid short cationic domain. The ACM is also a short cationic domain, which means mostly positively charged. And so these are on proteins or peptides. And what you see is when you force an interaction in between this low complexity RNA and this motif, you get the foci. And so you kind of correlate that these two interacting is enough for development of these condensations. And so what they also do is change the salt concentration of the solution that these reactions are taking in vitro and both in vivo. And what they find is there's a difference when you change the amount of salt in the reaction, even with the normal motif and the regular low complexity RNA. And so that points to there being an electrostatic interaction that is actually facilitating this interaction. And so to really pinpoint that, they make a little short 4-arginine peptide, all positively charged, and force an interaction with the low complexity RNA. And what that happens is you get these foci again. And so they really believe this interaction between this cationic positively charged domain and these negatively charged low complexity RNA is responsible for these foci for starting to develop. And then what they find is that um, once these foci develop, they're kind of more liquid-like initially, but then they start to harden. But the hardening is not actually caused by just the RNA and cationic domain interaction, but rather a fibrillation propensity domain. So basically, the RNA and cationic domain first interact, and this seeds the development of the phase separation, and that then activates a fibrillation propensity domain, which recruits more proteins and starts to rearrange them so that they begin to become folded into these beta sheets and really harden and solidify. And they can prove this because they do a frap. So in this frap, they bleach a little part of the foci, and what they find is you get instantaneous recovery without um, recapitulating any of the RNA or cationic domain reaction, but um, just the existence of the first initial seeding of the reaction is enough, because at that point, the fibrillation propensity domain has taken over and can allow for the expansion and hardening of the little condensate. And then also on C, you can see that a stress like acidosis also creates these bodies as well. And so to summarize this, basically what happens is a cell undergoes a certain cellular stress, and what happens is certain low complexity RNA starts to get transcribed, and this is negatively charged, and this interacts with the positively charged cationic domains on proteins to start to condense and come together. And this creates these first initial little liquid phase condensates. But then after that happens, the fibrillation propensity domain is activated, and then you start getting these proteins rearranged into these beta sheets, and they start to harden, and you get this physiological phase transition into the amyloid bodies. And so you may be thinking then, so, that's cool and stuff, but what's the point of this? And so I included this quote that was in the discussion. Um, a lot of pathological conditions, especially neurological conditions, uh, find that there are these amyloid bodies, plaques that start to develop around the brain, specifically in Alzheimer's. And so it may be very possible that these low complexity RNA regions are responsible for these plaques, and figuring that out may be important to how we may treat them. Um, and so, yeah, that's a really good application. And this paper was made in 2018, so I don't know if they've already kind of looked at this already. But also, what I think is really interesting about um, this kind of paper is it just kind of proves this idea that's kind of revolved around in our semester, that, nothing in the genome that's well conserved really is non-functional. Things that we used to think were just junk and did nothing actually are very important. And it's only then now, with advances in sequencing and microscopy and looking at different perspectives of sciences and combining them, that we realize that maybe these regions actually are functional in a different way than what we may normally think. And so that's why I really thought this paper was interesting. And yeah, so I hope you enjoyed it, and maybe it scratched your brain and made you kind of think about things differently. And so, yeah, these are just my references. And yeah, thanks for listening and watching.